Imagine, um, you know, two patients with major depressive disorder just sitting side by side in a clinic.
Right.
On paper, they have the exact same diagnosis. But when you actually talk to them, they share almost zero overlapping symptoms.
Oh, absolutely.
I mean, one can't sleep, right? They're intensely agitated. Maybe they have a racing heartbeat. And the other one sleeps like 14 hours a day, can barely move, and just feels this profound, heavy numbness.
Yeah, completely opposite experiences.
Yet the current medical paradigm often hands them the exact same pill and just, crosses its fingers.
Yeah.
Have you ever wondered why that antidepressant might work miraculously for the first person, but do absolutely nothing for the second?
It's a huge problem. And it really comes down to a fundamental misunderstanding of the diagnosis itself. We treat major depressive disorder, or MDD, as a single monolith, but it's actually an umbrella term.
An umbrella for like a bunch of different things.
Yeah. Exactly. Underneath that umbrella is a wildly diverse collection of biological misfires. And because the diagnostic criteria are so broad, finding a single universal treatment is mathematically and biologically impossible.
We're basically throwing a single wrench at a dozen different types of broken engines.
That's a great way to put it. We've been doing that for decades.
Which is exactly why we are pulling apart a truly fascinating June 2026 paper from Nature Genetics today. Welcome to this deep dive. The researchers behind this study didn't just look at the symptoms of depression. They actually mapped its genetic architecture inside a living brain using a groundbreaking technology called in vivo Perturb-seq.
And the stakes here are incredibly high. For over 20 years, we've had tools that could point out which genes are associated with depression. But a list of genes isn't a mechanism.
Right, a list is just a list.
Exactly. We need to know why those specific genes matter. How do they actually disrupt the brain's circuitry in real time to cause a depressive state? And what we're going to explore today uncovers a highly specific cluster of genes that point to a shockingly treatable biological pathway.
Okay, I want to get right into the mechanics of this, because historically, psychiatry has heavily relied on genome-wide association studies, or GWS.
Yes, GWS has been the standard.
Right. And GWAS involves taking the DNA of hundreds of thousands of people and scanning it to find genetic variations linked to a disease. And it's been incredibly successful at giving us a list of risk genes for depression. But it just stops there.
It does. It gives you the what, but not the how.
To me, it feels a bit like looking at a fuzzy satellite image of a city at night. You can see that a certain neighborhood has lost power, but you have no idea if a transformer blew or a wire was cut or someone just forgot to pay the bill.
That analogy captures the limitation perfectly. So the researchers for the study, they started by sifting through 7 different major GWS databases. They took that broad satellite imagery and cross-referenced it with high-resolution bulk RNA sequencing data from actual human patients.
Real patient data, okay.
Yes. And they focused their lens on one specific anatomical region, the dorsolateral prefrontal cortex, or DLPFC.
The DLPFC, that's essentially the brain's executive control center, right? It regulates our emotions, our motivation, our complex decision-making.
Precisely. When you are assessing whether a task is worth the effort, that's your DLPFC at work.
And when it goes offline, the deeper emotional centers of the brain just run wild.
Right. So within the DLPFC of human patients with MDD, the researchers found 79 distinct risk genes that consistently showed lower expression.
Lower expression meaning?
Meaning they were physically present, but they were turned down, underperforming. Now, because the actual experimental phase of this study needed to be conducted in mice, the team carefully matched those human genes to their exact mouse equivalents.
Which narrowed the suspect list down, I assume.
It did. It narrowed it down to 44 target genes.
Okay, so we have 44 genes that we know are intimately involved in human depression. Now, in the past, if scientists wanted to know what one of those genes did, they'd either study it in a petri dish or spend years breeding a genetically modified mouse missing just that one specific gene.
Which is incredibly tedious.
Right. And a petri dish doesn't have the electrical complexity or the diverse cell types of a living brain. And breeding 44 different custom mouse lines, that would literally take a lifetime.
Which brings us to the massive technological leap here. They used in vivo AAV-Perturb-seq. So instead of taking cells out of the brain or breeding decades worth of mice, they edited the cells directly inside the brains of living, breathing adult mice.
This mechanism is wild to me. Let's break down how you actually hack a living brain cell. They used adeno-associated viruses, or AAVs, right?
Yes, specifically a highly specialized variant called PHP.EB.
PHP.EB. Why that one specifically?
Well, the challenge with brain research is the blood-brain barrier. It's this microscopic, highly selective fortress designed to keep pathogens out of the central nervous system. Most traditional viruses or drugs just bounce right off.
I can't get in.
Right. But the PHP.EB viral vector is unique. Think of it like a biochemical smuggler that has acquired the exact forged credentials needed to slip right past the guards.
A smuggler. I like that. And once this smuggler gets inside the brain, it has to deliver a payload. The payload here was a library of dual CRISPR scissors, single guide RNAs.
Exactly.
So they injected millions of these viral smugglers directly into the bloodstream of female Cas9 mice. Let's pause on the biology of these mice for a second, because the setup is brilliant. First, they used female mice.
And that wasn't arbitrary.
Right. Major depressive disorder is significantly more prevalent in human females, so the model really had to reflect that specific neurobiology.
Exactly. And second, they are Cas9 mice. This means the mice were already genetically engineered from birth so that every cell in their body naturally produces the Cas9 protein.
Right, and Cas9 is the engine, like the actual cutting motor of the CRISPR system.
Yes, exactly.
So the researchers didn't have to pack the heavy cutting machinery into the viral smuggler. The cells already had the scissors, they just didn't know where to cut.
Spot on. The virus merely delivered the GPS coordinates, the guide RNAs, telling the Cas9 motor to seek out and knock out one of those 44 suspect genes.
But wait, once you inject this into a living animal, you have a massive chaotic environment. How do you know which neuron received which guide RNA?
To solve this, the viral payload also included a tracking device, a nuclear localizing M-cherry protein.
M-cherry. So it makes the nucleus of the cell literally glow red under a microscope.
Yes, literally glows red. So they let the mice live normally for about 4 weeks. This gave the CRISPR system time to edit the DNA, and more importantly, it gave the brain's chemistry time to alter in response to that missing gene.
Right, because you need to see the fallout.
Exactly. Then they extracted the brains, sorted out all those glowing red nuclei, and ran single-nucleus RNA sequencing on them. They sequenced an astounding 169,186 high-quality individual nuclei.
Almost 170,000. That's massive. By doing single-nucleus RNA sequencing, you are basically opening the factory floor of the cell and reading every single biological receipt. You can see exactly which proteins are being manufactured and which assembly lines have shut down.
And there's a vital mathematical concept here that makes this data usable. MOI, or multiplicity of infection.
Oh, right. Explain that for us.
This is critical for the precision of the study. If a single neuron takes up 5 different viral vectors and 5 different genes get knocked out, the resulting data is completely useless. You wouldn't know which missing gene caused the cellular collapse.
It would just be noise.
Exactly. So they carefully titrated the viral dose to ensure an MOI of 1. Statistically, they filtered the data so they were only looking at cells that took up exactly 1 guide RNA. 1 cell, 1 targeted genetic knockout.
Okay, let's just do a quick recap of where we are. So, bullet point 1, we have our list of 44 suspects based on human data.
Yep.
Point 2, we have a fleet of viral smugglers that slip past the blood-brain barrier.
The PHP.EB vectors.
Right. Point 3, they delivered highly specific CRISPR coordinates to thousands of individual cells in female Cas9 mice. And point 4, they left a glowing red M-cherry tag so we could sort them and read the biological fallout with an MOI of 1. It's an unprecedented level of control.
It really is.
I mean, if you had a microscopic smuggler that could safely edit one specific type of cell in your body, what would you choose to fix? It's a profound thought experiment, but it also highlights the danger of this kind of intervention. Because you can't just go in, delete random genes, and expect the brain to shrug it off.
Absolutely not. The cellular ecosystem is incredibly fragile, and the immediate effects they observed really prove that. Out of the 35 distinct cell clusters they analyzed in the brain, they found that certain genes are essentially structural load-bearing walls.
What happens when you knock those out?
Well, when they knocked out genes like ERK2, ETV6, or ANO10, the cells just couldn't cope. The result wasn't a behavioral change, it was just a massive drop in the survival rate of those neurons.
The neurons literally died.
Yeah.
Which makes sense. I mean, past literature shows these genes are critical for basic physiological survival, like neuromuscular function. If you take out the foundation, the house collapses.
Exactly. But the researchers weren't looking for genes that cause cell death. MDD isn't characterized by massive neuronal die-off. It's characterized by subtle, devastating shifts in function.
Right.
The genes responsible for depression don't act like wrecking balls. They act like a badly tuned instrument in an orchestra, throwing off the entire symphony. Most of these 44 genes are not direct transcriptional regulators.
Meaning they don't just act as simple on or off switches for other genes. So if they aren't the switches, how do you measure what they are actually doing?
You have to map the indirect consequences. To do this, the team used a highly advanced computational framework called HDWGCNA.
Let me guess, a huge acronym.
Yes, that stands for High-Dimensional Weighted Gene Co-Expression Network Analysis.
Okay, let's untangle that a bit. What does that actually look like?
Think of it this way. Imagine the metabolic pathways of a cell, like a massive three-dimensional spider web. If you perturb just one ion channel on the outer membrane of a neuron, you alter the way calcium flows into that cell.
Okay, calcium changes.
Right, and that subtle shift in calcium changes how the cell metabolizes energy, which in turn alters the RNA readouts of hundreds of completely unrelated genes deep inside the nucleus. HDWGCNA is a mathematical model that tracks the covariation of all these thousands of genes.
So it's looking at the whole web shaking.
Exactly. It maps the cascading ripple effects, allowing researchers to see how knocking out one poorly understood risk gene distorts the entire functional network of the neuron.
So they mapped the unique ripple effect for all 44 genes in the mice. But an isolated data set doesn't mean much until you cross-reference it with reality. So they took these 44 distinct mass crime scenes and held them up against actual transcriptomic data from the brains of human female MDD patients. They essentially asked the algorithm, which of these mouse models actually looks like the human brain struggling with depression?
And the result was a massive breakthrough for psychiatric genetics. Based on the similarity of their ripple effects, the 44 genes grouped into 5 distinct modules, or clusters, and only one of those clusters perfectly mirrored the specific cellular breakdown seen in the human MDD brain profile.
Just one cluster out of 5.
Just cluster 1. It contained a tight-knit group of exactly 6 genes: DEN1A, ERB4, NKN2, TOX, SNRK, and PZLO2.
Wow.
Yeah. When any one of these 6 genes was deleted in the mouse, the resulting network ripple showed a severe downregulation in neuronal development and synaptic signaling. The neurons just weren't communicating effectively, and that matched the human patient data flawlessly.
I have to pause and push back on the scale of this finding. We started with 44 widely accepted, genetically verified depression genes found in massive human databases. You're saying that only a tiny gang of 6 actually recreates the specific transcriptomic signature we see in these particular human patients.
Exactly. And this circles back to the very first thing we discussed, the heterogeneity of depression.
The umbrella term.
Right. What about the other 38 genes? They're still statistically linked to MDD, but their loss creates entirely different biological ripples. They likely represent completely different biological subtypes of depression that modern psychiatry hasn't even begun to properly classify yet.
That's staggering.
It is. But for this specific observed patient profile, cluster 1 was the Rosetta Stone. And within that cluster, the DEN1A gene was the absolute star, showing the highest mathematical similarity to the human disease state.
Okay, let's quickly recap this second part before we move on. So the team didn't just knock out genes. They built a mathematical model of the metabolic ripples spreading through the cells, right? And they discovered that only the 6 genes in cluster 1 recreate the exact human MDD signature.
Perfectly summarized.
Consider how intricate your own consciousness is, right? The fact that deleting one single gene in a mouse can perfectly mirror the complex network collapse of a human psychiatric condition is just mind-bending.
It's a triumph of molecular mapping. But, as we know, a map doesn't cure the disease. The researchers had identified the 6 culprits, but they still needed to understand the common denominator. What specific biological mechanism made cluster 1 unique compared to the other 4 clusters?
It had to be a shared pathway, right? Like a missing molecular link.
It was. The team ran a gene set enrichment analysis, which is basically a way of looking at all the disrupted pathways to find the loudest signal. They discovered that the loss of any of these 6 cluster 1 genes uniformly led to the severe suppression of the oxytocin signaling pathway.
Oxytocin. Now, most people know it as the love hormone or, the bonding molecule released during childbirth or hugging.
Yeah.
But its role in the brain is much more mechanical than that, isn't it?
Much more. In regions like the DLPFC, oxytocin acts as a critical neuromodulator. It tunes the signal-to-noise ratio.
How so?
Well, it interacts with specific inner neurons to lower their threshold for firing, which regulates how the brain processes synaptic signals regarding effort, reward, and motivation. Without oxytocin signaling, that entire circuit just goes dark.
And the specificity here is what makes this paper such a huge deal. This oxytocin collapse was exclusively specific to cluster 1. Like if they knocked out a well-known MDD risk gene from a different cluster, say LHPP, the oxytocin pathway was completely untouched.
Exactly. The engine was broken, but the oxytocin spark plugs were fine.
Furthermore, they validated this in humans, didn't they?
They did. They looked at a cohort of individual human MDD patients who specifically exhibited lowered oxytocin signaling in their neurons. And in those exact patients, 5 of the 6 cluster 1 genes were found to be heavily suppressed. The correlation was rock solid.
It's incredible to see the molecular pathway turn off on a spreadsheet. But, biology isn't just spreadsheets. Does turning off that pathway actually make the animal depressed?
That's the ultimate test.
Right. So to prove it, the researchers moved from sequencing to behavior. They took mice and knocked out just the tox gene or just the DEN1A gene.
And the behavioral phenotype they induced was really striking.
And keep in mind, they didn't subject these mice to any external stressors. There was no unpredictable environment, no isolation, no physical stress. But just by knocking out that one gene, the mice developed profound depressive-like behaviors. They used 2 standard assays for this, right? The tail suspension test and the forced swim test.
Yes. These tests measure a very specific psychological construct, behavioral despair. It quantifies a mouse's internal drive to keep struggling in an inescapable situation.
Exactly. And the mice missing the tox gene exhibited significantly increased immobility. They just gave up quicker. Instead of swimming and looking for a way out, they just floated.
Which perfectly models the anhedonia, the deep lethargy, and the sheer loss of motivation we see in human depression.
They induced a massive psychiatric collapse by turning off a single node in the genetic network. But this is where the paper delivers its most crucial finding, the rescue.
Yes, the rescue is fascinating.
If the oxytocin pathway is the thing that broke, can you just manually replace it?
Well, they tried. The researchers took these tox-deficient, unmotivated mice and treated them pharmacologically with oxytocin, as well as a longer-lasting, more stable analog called carbetocin.
And what happened to the circuitry?
The depressive behaviors were partially alleviated. The mice actually started swimming again. Their drive to try returned. By providing a pharmacological hit of oxytocin, they bypassed the broken genetic switch entirely. Yeah, they artificially turned the downstream circuit back on, restoring the synaptic tone in the prefrontal cortex. The motivation returned because the chemical currency of effort had been restored.
I look at a result like that and I immediately think, can we just start prescribing oxytocin nasal sprays to everyone who walks into a clinic with a diagnosis of depression?
That is the temptation, definitely. But the answer is a definitive no. And understanding why the answer is no is probably the most important takeaway of this entire study.
Why not? I mean, if it restored the behavior in the mice.
Because of patient stratification. Past clinical trials have actually attempted to use oxytocin as a broad treatment for psychiatric disorders, including autism and depression, and they largely failed.
Why did they fail?
They failed because the researchers gave the drug to everyone with the umbrella diagnosis. But this paper mathematically proves that oxytocin won't cure all depression, will only work for the specific subtype of patients who have a dysfunction in those specific cluster 1 genes.
So if your depression is being driven by one of the other 38 genes we talked about, giving you oxytocin won't do a thing. Because your oxytocin pathway isn't the one that's broken. You have a different flat tire entirely.
Precisely. This study proves that depression has distinct, treatable biological subtypes that require entirely different pharmacological keys.
Think about how we approach physical ailments today versus mental ones. In oncology, you don't just get a generic bag of chemo anymore. A doctor biopsies the tumor, sequences its DNA, and gives you a targeted therapy designed for your exact genetic mutation.
Which is how it should be.
Right. Could the future of psychiatry be that personalized? Imagine a world where you don't just get a trial and error antidepressant, but a highly targeted molecular rescue engineered for your specific genetic short circuit.
That is exactly the paradigm shift we are looking at. We are finally moving from diagnosing the symptom to diagnosing the circuit.
Thank you for joining us on this intensely detailed deep dive. We started by looking at a fuzzy satellite map of a GVORS database. We wrote a microscopic viral smuggler through the heavily guarded blood-brain barrier. We mapped the mathematical ripples of a single edited cell. And we ended up restoring a mouse's will to swim by artificially bypassing its genetic code with oxytocin.
It's a truly remarkable journey. And I want to leave you with a final paradigm-shifting idea to mull over. We've just established that major depressive disorder isn't a single disease, but a collection of completely distinct genetic short circuits. What does this mean for the rest of the DSM?
Oh, that is a massive question.
Right. Think about conditions like anxiety, ADHD, schizophrenia, or bipolar disorder. Are they also massive umbrella terms hiding completely distinct biological subtypes? Are there millions of patients right now suffering through side effects from broad-spectrum treatments when their specific biological subtype is just waiting for its own unique, highly targeted molecular rescue?
It really forces you to look at every medical diagnosis a little differently. Suddenly, that clean, simple X-ray of a broken arm doesn't seem so vastly superior to the murky waters of psychiatry. We just needed a better molecular flashlight to see through the mud. Thanks for listening, and we'll catch you on the next Deep Dive.